So, hi everyone. It's my pleasure to introduce Windie Hoefs. I hope I said that correctly. For today's Future Leaders in Mechanobiology Symposium. So Windie carried out her undergraduate degree at UCL in London. And then she joined the Francis Crick Institute for her PhD, where she was co-supervised by Dr. Nicolas Tapon and Guillaume Charas. And in their lab, she studied the translocation dynamics of YAP protein. I know many people here care a lot about YAP, so we're really looking forward to hearing more about it. And she particularly studied the role of E-cadherin in that translocation. She just told me that, so she finished her PhD recently, and she just started a week ago her postdoc in Sarah Wikström's lab in Germany at the Max Planck Institute, where she'll continue studying how mechanical forces impact cell fate in hair follicle cells. She's won several awards, in particular a Collaborating Creatively Annual Crick Award for her PhD work. We're really looking forward to hearing more about that work at today's talk. So Windie, without further ado, I give the platform to you.
Thank you so much. I hope everyone can hear me. Thank you for inviting me today to the Future Leaders in Mechanobiology seminar. I'm really excited to present you the work that I've done in my PhD. So just as a brief introduction, as you all know, cells can sense physical forces. And let me just quickly change to the laser pointer. And this force sensation and response is crucial in various biological processes, such as in physiology, development, as well as disease. Okay. For example, in physiology, as you know, as we breathe and our lungs expand and contract, the cells aligning the lungs also have to stretch. And this is important for the organ function. Next, also in development, forces that cells can sense are important for the role, such as in developmental transitions. For example, in the Tapon lab recent research on the growing fly abdomen, which is a histoblast nest, we can see that basically the two main cell types, the larval cells, which are the big cells in the movie, display different physical properties to the adult cells that become basically the adult abdomen. And these are important to have different physical parameters for the different cells for a robust transition to occur. Last but not least, also in disease, physical forces play an important role. For example, the Saha lab at the Crick has shown previously that in the breast cancer tumor niche, the surrounding healthy cells, the fibroblasts, they can, over time, gain a more cancer-aggressive phenotype through these cells sensing the increased ECM stiffness as more collagen is deposited as the tumor niche grows. And cells can sense physical forces mainly through key specific protein complexes, which are called the mechanotransducers. So these proteins basically change the conformation upon force sensation and allow the signal to be relayed to the internal part of the cell. And I'll go briefly into each category. So starting off with the focal adhesions. The focal adhesions, as you know, are the receptors at the basal level of the cell that connects the cell to the substrate and can sense the changes in the physical properties of the substrate. For example, upon ECM stiffening, the receptors, the integrins, are cluster forming the focal adhesions and can in turn recruit the adapter protein talin, which unfolds and allows further recruitment of the vinculin, ensuring that the force is relayed to the internal part of the cell via the cytoskeleton, the F-actin. Next, the pushing and pulling of cells can be sensed through the cell-cell junctions, mainly the adherent junctions, which are known to bear basically the mechanical load. And the adherent junctions are Sorry, I just saw the chat. I thought it was a question for me. So the adherence junctions are composed of the eka-adherent homophilic bond, and in turn, they can again relay the force to the internal part of the cell through being connected to the F-actin via the adapter proteins beta and alpha-catenin. Last but not least, also the cytoskeleton acts as a mechanotransducer through relaying the force signals that come from the focal adhesions and cell-cell junctions, but also through producing intracellular forces through the actomyosin contraction. And together, these three mechanotransducers can individually sense the forces and relay them into the internal part of the cell, where the force is basically converted to a biochemical signal, allowing the cells to adapt their gene expression in order to conform to the changes in the physical environment. And this process is called mechanotransduction. And a key protein involved downstream of mechanotransduction, as you all probably know, is the YES-associated protein, YAP. So YAP in mammalian cells is known to be mechanosensitive. With this, I mean, upon mechanical stimuli, it can be stimulated to translocate into the nucleus, where it binds to transcription factors T1 to 4 to promote cytoskeletal, pro-growth, as well as self-renewal genes. For today, I'm just going to mainly focus on the mechanical stimuli that affect YAP via the two key mechanotransducers, the focal adhesions and cell-cell junctions. So starting off with mechanical stimuli affecting mainly the focal adhesions and in turn also YAP. Plating cells on different substrate regimens, so rigidities, affects YAP localizations. For example, when mesenchymal stem cells are plated on a soft substrate, YAP is known to be more cytoplasmic, which induces adipocyte formation. In contrast, when mesenchymal cells are grown on stiff substrates, it promotes osteogenic differentiation and induces YAP nuclear localization. This is also due to, at stiff rigidities, focal adhesion maturation being promoted. Similarly, plating cells on different substrates areas can also affect YAP localization. For example, plating cells on a large substrate leads to cells being able to form more integrin connections and therefore leading to YAP being in the nucleus. Compared to focal adhesion, the cell junctions have a more negative regulation in YAP localization. This can be seen as at high cell density, where there are a lot of cell-cell junctions, YAP is cytoplasmic. In contrast, at low cell density, where cells can spread and with this have more cell ECM availability, YAP is nuclear. Interestingly though, a study has shown that this high density negative regulation of YAP can be reverted when strain is applied. And I'll go a little bit further in detail now about this particular study. In the past, a study has shown that when plating a well-known epithelial cell line in the biophysics field, the MDCK, maden-darby-canine kidney cells, when these cells are plated at high cell density on a deformable substrate, then YAP is cytoplasmic. And this makes sense as here there are a lot of cell-cell junctions which are known to be negative regulators. But when biaxial strain is applied, after 1 hour, this mixed localization is shown in this immunostain of the MDCKs. And mixed localization of YAP is indicative in the MDCK field of nuclear YAP. The authors have concluded in this particular study that the strain-induced YAP nuclear localization is mainly due to the cell-cell junctions being stretched. However, we can appreciate from the testing system that the cell ECM connections are also present and therefore may also influence YAP localization. Indeed, more recent single molecule studies have shown that when stretching cells on a deformable substrate, the components of the focal adhesions are also getting stretched, as shown here with the Paxillin GFP staining. Therefore, from these studies, my major question for my PhD was to understand what the distinct roles are of the cell-cell junctions and focal adhesions in YAP mechanolocalization. And to do so, I've developed 3 assays, which basically tests the individual roles of the cell-cell junctions and focal adhesions in YAP localization at different dynamics. So I looked at monolayer growth, dissociation, and stretch. Starting off with monolayer growth, here my major aim was to understand how YAP in real-time localization changes as the cell-cell junctions increase. For this, I've developed throughout my PhD and using CRISPR-Cas9 an endogenously tagged YAP MDCK line using the halo tag as a tag. And what we can see from the generated cell line is that it follows basically the characteristic cell density dependent localization of YAP, With this, I mean at low cell density, YAP is mixed, so basically nuclear, leading to a high nuclear to cytoplasmic ratio as quantified here. While at high cell density, YAP is excluded from the nucleus, and this is also shown with the low nuclear to cytoplasmic ratio quantified. From this result, we know that the presence of ecadherin affects YAP localization. We were further wondering whether also the protein levels of the cell-cell junctions to the ecadherins affects YAP localization. And to do so, I've developed an overexpressed ecadherin line. Live imaging of the overexpressed ecadherin line has shown that the moment the first junction is formed, YAP is already immediately excluded from the nucleus. And further closer inspection visually, you can see the comparison. So at low cell density, we again see very cytoplasmic YAP compared to the endogenous ecadherin protein level line, where YAP is more mixed or more nuclear. And indeed, quantification of these two lines has shown that at low cell density, YAP nuclear localization is reduced in the cell line where the ecadherin levels are higher. However, interestingly, at high cell density, there's no significant difference. So again, this could be a thing that ecadherin levels and presence are more important in reducing YAP nuclear localizations throughout monolayer growth. But the moment a high density is reached and YAP is already excluded, then the protein levels don't matter anymore because YAP is already outside of the nucleus. Therefore, from my characterization of the endogenously tagged YAP MDCK line that I generated, we can see that as the cells are growing to form a monolayer, YAP is excluded from the nucleus, and this is in part due to the levels of cell-cell junctions as well as the presence. From this study, we further wanted to understand, well, can this effect be reverted? So can we decrease cell-cell junctions? What happens if instead of looking at monolayer growth, we look at dissociation? This basically leads to my second essay, which is looking at dissociation of cellular aggregates using EMT, epithelial to mesenchymal transition. So a brief overview, epithelial to mesenchymal transition is when epithelial colonies basically start to have the cells dissociating over time. And it is a key process in, for example, cancer metastasis, where basically the cells start to dissociate and spread throughout the body. In this process, the polarity markers are lost and the cells become more and more flat that have torn apart from the colonies, becoming mesenchymal. And we were wondering whether the reduction of the cell-cell junctions as the polarity markers are being lost could affect YAP localization. Indeed, we know from past studies that YAP induces EMT. And this is shown, for example, here, where with the MCF10A cells, when YAP is overexpressed, we see this rescattering effect and this more aggressive phenotype reminiscing of what could happen in cancer metastasis. But in past studies have not really looked at YAP localization. They only looked at this phenotypic effect of YAP. Therefore, using the generated cell line that I made of endogenously tagged YAP and MDCK, it was a good avenue to further analyze YAP localization role in EMT. It is known that MDCKs can be induced to undergo EMT by treating the cells with the hepatocyte growth factor. So I tested this in my line, and we could see that 1 hour treatment with HGF did not seem to change the localization of YAP, and also an immunostaining against the ecadherin antibody, you could see that there's no real change in the organization of the ecadherin. However, upon prolonged treatment with HGF, we suddenly see mixed localization of YAP, and the cells are much more deformed than the control and bigger. Indeed, also the ecadherin organization is lost. The cells don't form anymore this rigid symmetry. They are more wiggly, indicative that there's a perturbation in the ecadherins. And indeed, further live imaging of the cells upon HGF treatment shows that they are induced to scatter. And quantification has shown that there is an increase in YAP nuclear to cytoplasmic ratio over time in the HGF-treated cells compared to the control. And this basically marks the onset of the spreading. Looking at individuals' time points and the scatter of the nuclear to cytoplasmic ratio further showed to us that there is a significant increase in YAP nuclear localization upon induction of EMT. And interestingly, this localization also increase in localization also persists all the way to the later time points. However, there is also slightly an increase in the control, and this could be because in both setups, the cells were not grown to full confluency. So in the control, they were still able to spread, leading to a bit of nuclear YAP still being around. So further studies would need to further look at taking live imaging movies that were of longer duration to see whether at some point also there's a further decrease in the HGF-treated YAP nuclear to cytoplasmic ratio. But overall, what we can see from our EMT study is that by inducing EMT in the MDC case, there seems to be an increase in nuclear YAP that persists for a long time. And this could be in part due to the reorganization of EKD and the perturbation that the cells are able to scatter, meaning that they have more ECM availability. But we also know that when MDCKs are treated with HGF, a past study has shown that this also leads to more contractility that involves the focal adhesions basically pulling on the cytoskeleton, which allows the cell junctions to tear apart. So we were wondering, since focal adhesions seem to have an impact in EMT, whether this could also affect YAP localization. To check this, I've indirectly treated the cells with downstream focal adhesion signaling inhibitor, which is SAR kinase. So the inhibitor blocks SAR kinase, and it's called dasatinib. So initially, I just wanted to check whether the inhibitor works in the cell line generated. And indeed, what we can see is that at low cell density, where YAP is more nuclear, upon dasatinib treatment, where the SAR kinase pathway is blocked, which is known to, when active, upregulate YAP into the nucleus by phosphorylating YAP at tyrosine 357. We can see that the YAP localization is more cytoplasmic, and it's also been quantified. Similarly, pre-treating cells with HGF so that they undergo EMT and then adding dasatinib also led to a decrease in YAP localization in the nucleus. And what was quite striking is that this localization is similar to the basal condition where no treatment was added. So this study suggests that possibly the SARC signaling is key for promoting YAP nuclear localization during EMT, along with the cell junctions being broken down. So now that I've further characterized the contributions of both the cell junctions and the focal adhesions in YAP localization, I wanted to understand what occurs to these two mechanotransducers in regulating YAP localization in a dynamic system such as upon stretch. So since I have a tool to look at live YAP, what we wanted to further understand is whether this conclusion from the past study I've introduced before is true, whether stretching solely the cell junctions leads to YAP nuclear localization. To do so, I used two different stretching systems. I'm going to introduce the first one now. So the first stretching system was looking at stretching both the cell junctions and the cell ECM. So this is basically a modified version of the Charras stretching device at UCL that I've developed. So the stretching device consists of two glass cover slips shown here. And in between these two glass cover slips, there's a deformable substrate shown in gray, which is PDMS. Then the two glass cover slips are held together by a bent glass capillary. And note that this right side of the glass capillary is flexible. Next, for the cells to grow on this deformable substrate, I have to add collagen. The collagen is dried, and then for 1 to 2 days, I incubate my MDCK cells on top of this collagen to form a robust monolayer across the two glass cover slips. Then, because this deformable arm is present, I'm able to pull on this arm to obtain a uniaxial strain, which can be visualized in an inverted microscope. And what we can basically see when applying 12.4% strain is that over an hour time, relocalization occurs. It will loop again, so then you can see. So initially, YAP is excluded from the nucleus, that's a black hole. But then the moment after stretch, we obtain this relocalization, this mixed localization over time. So for me, this result was quite exciting because it recapitulates in a slightly different stretching system the published work I've introduced before, that upon stretching MDCK cells on a deformable substrate, we obtain after an hour a relocalization of YAP into the nucleus. And to test whether this is truly due to strain, I also performed control monolayer experiments where I seeded the cells on the PDMS but did not pull on the arm, so there was no strain. And what we can see indeed is that in those conditions where zero strain was applied, YAP localization remains cytoplasmic. The black hole referring to the nuclei remains over time, over the 1 hour imaging. So this further proves that indeed it seems that in the presence of both the cell-cell junctions and the cell ECM context, when strain is applied, YAP goes into the nucleus. But this is nothing new. We already mentioned it before. So the next stage to really dissect which mechanotransducer, or if both are really important for strain-induced YAP nuclear localization, the next step was to understand, okay, what happens to YAP localization when there's no substrate? And with this, strain is only applied to the cell-cell junctions. To do so, I've used the stretching device again, but it's slightly different. If you note carefully, this time there's no gray layer, there's no deformable substrate, there's no PDMS. Instead, the first substrate added is the collagen. Here, the collagen acts as a sacrificial substrate, in that after the collagen is dried and I seeded my cells and they form a monolayer, I then apply collagenase treatment. The collagenase digests away the collagen, leaving me then with a monolayer lacking substrate. It's devoid of substrate, forming a suspended monolayer across the 2 glass cover slips. Again, through applying uniaxial strain by pulling on this flexible arm, I'm able to then visualize the stretched monolayer on a microscope, and this time applying a higher strain, because since there's no rigid PDMS, no deformable substrate, I can stretch more the monolayer. We can see that over time, there's no relocalization occurring. So in the absence of cell ECM contacts, the strain-induced YAP nuclear localization does not occur. Further proving, and strikingly, that it seems to be mainly the cell ECM being stretched that induces YAP nuclear localization. To further test this hypothesis, my more recent experiments were to check whether it's the integrin downstream signaling affecting the localization of YAP in stretched monolayers where the substrate is present. And to do so, I basically used again this indirect treatment, which was applying dasatinib treatment, which blocks basically the downstream focal adhesion signaling that usually promotes YAP nuclear localization. So I thought by blocking this signaling, by adding dasatinib on a monolayer with PDMS, maybe I could change the before nuclear localization that we expect. To do so, what I did is pre-treat my monolayers with dasatinib before I applied stretch at a similar regimen where I usually see nuclear localization. So you can see that by 11.3% strain after dasatinib treatment, we do not get a relocalization. So inhibiting SARC signaling blocks this stress-induced YAP nuclear localization. You can see over time, the YAP remains excluded from the nucleus. In conclusion, from my work, we can see that there seems to be an antagonism between the cell-cell junctions and the cell ECM in regulating the YAP and epithelial aggregates. From the monolayer growth work I presented earlier, we can see that as the cells increase in number and form cell-cell junctions, the YAP is excluded from the nucleus, and this is mainly due to the cell-cell junctions playing a more dominant role as they increase in number. In contrast, though, at this stage, the cell ECM availability throughout monolayer growth decreases as the individual cells, as they form the colonies, experience less and less cell ECM contacts. The opposite occurs then when looking at the EMT system, where I basically induce the cells to decrease in cell-cell junctions. And what we can see is that by perturbing the cell-cell junctions, YAP nuclear localization is promoted, and as the cells scatter and individually then experience more cell ECM contacts, the cell ECM signaling becomes more important. And this is what we saw also with the dasatinib SARC signaling inhibition, that it is key for blocking then the nuclear localization in EMT. And last but not least, from my stretching work, we can see the striking result that it seems that it's mainly stretching the cell ECM contacts that promotes YAP nuclear localization, because stretching monolayers devoid of cell ECM contacts led to YAP remaining cytoplasmic. And then last but not least, with my dasatinib treatment on the monolayers that did have substrate, and this also blocking YAP localization into the nucleus, it further shows that it seems to be the cell ECM along with the downstream signaling that is important for stress-induced YAP nuclear localization. From this body of work, it further gives an idea that it seems that there's an emerging role of ECM remodeling that could affect YAP regulation in general. For example, a more recent study has shown that in patients that have a relapse in ovarian cancer, there is a change in the deposition of collagen, in that there's an increase in collagen 6, which also correlates with more resistance to chemotherapy. And one reason could be, I'm just hypothesizing here, is that possibly this change in ECM composition could affect, for example, pathways such as YAP and induce it to be more active, which could lead it to more aggressive cancers that are chemo-resistant. Also in development, ECM remodeling plays a role. And here I would like to also shout out to my colleagues in the Tapon lab with the recent paper on the fly abdomen development and the biophysics about it. Basically, my colleagues have shown that for a robust abdomen to form, the ECM must be degraded in the Drosophila pupil abdomen. And preliminary work has also shown that this ECM degradation is also accompanied with the target gene expression of the YAP ortholog Yorkie being decreased. So possibly what regulates the whole time YAP in all these different systems in disease as well as development could be the ECM remodeling that changes the activity of YAP and in turn allows then the transitions in disease and development to be modulated and tightly regulated. But this is basically the next open question. With this, I would like to say thank you to both labs, the Charras lab at UCL, here we go here, and the Tapon lab at the CRIC for all of their help and their feedback. And it has been super fun working in both labs. I would also like to say thank you to the science technology platforms at the CRIC and UCL for helping me develop all the reagents I needed for my PhD project, my thesis committee members for all of their feedbacks throughout the process, and all of you for attending today, as well as the organizing committee for the seminar. It has been a fun day for me meeting the rest of the faculty. And as introduced before, I will be from next week onwards in the Wikström laboratory and hope to meet all of you at some point in a mechanobiology conference.
That was a wonderful talk, Windie. Thank you so much. I know that there are questions already in the chat box, so I'm gonna call out people to unmute themselves and ask their questions. So Narayanan is first. Are you still here and can you unmute yourself and ask your question? I can also read it out.
I saw here.
So the question was, is there any role of the beta-catenin in the dialogue between ecadherin and YAP?
That's a really good question. So in the salsa junction literature and how it regulates YAP, people were mainly focused actually on alpha-catenin more than beta. But what is known is that if you perturb the ecadherin junction, so that's like with the alpha-catenin, like the whole adherent junction, which is also with the alpha-catenin and beta-catenin is showed, then it does lead to YAP not relocalizing in the cytoplasm. So it is important to have a stable adherent junctions. However, I must say also then in the Drosophila work, some people have shown that if you mutate some bits and pieces from the adherent junctions, it can lead to other changes in YAP. So it's very system to system dependent.
I see. I know Dennis has a bunch of questions. Dennis, do you want to unmute yourself? I don't know if he's still here. He was wondering.
Hello?
Oh yeah, there we go.
Sorry. Yeah, so I wrote a question about YAP, entry into the nucleus seeming heterogeneous. So to me, I don't know if you quantified heterogeneity. Many of your nuclear over cytoplasmic, distributions look rather broad, but typical for the field. But do you know whether heterogeneity reflects heterogeneity at the single cell level in the strain levels? You apply it globally. So
That's a really good question. So as in, like, did I quantify the individual strain of the cells, like the individual cells? Is that
Yeah.
what you meant?
for example, or maybe you have other ideas. Maybe, for example, it's cell cycle. Maybe it's something else. But anyways, do you have an idea what the heterogeneity?
What it is. So basically, I haven't quantified it yet because I just got all the data for looking at the really individual cell heterogeneity, but there is. So I did quantify in general and to see what the YAP nuclear to cytoplasmic ratio is of individual cells. And it's really like, it's a scatter, like it's a distribution. But I think where it could be, and it has been also reported recently, actually, I'm not sure if it's a bioRxiv paper or a published paper, really sorry to the others that I don't know, by I think it was the Benoit Ladoux lab that have recently also looked at MDCKs, or in collaboration with the Benoit Ladoux lab, and they showed that it's to do with like the nuclear shape. So that could be a thing too, that it's like based on how like flat and the nucleus is to the extent to which YAP is inside, which is also something that the Pero Roca Cusas lab has alluded to a couple of years ago with the work from Alberto Artola Ilosogi.
Uh-huh.
That makes
And
sense.
I don't know if there's time or maybe later. The endogenous, looking at the endogenous is a great idea, but I'm also the Allen Institute, if you know or not, this Allen Institute for Cell Biology in Seattle, right, has done a lot of endogenous tagging, GFP, not halo tagging, and had a nice paper several years, maybe 4 or 5 years ago already. At the time, they had a dozen or 2 dozen, GFP, RFP, endogenous tags, generally single alleles, but showed that it was affecting levels, generally suppressing levels of expression. It's a great idea to have the endogenous promoter and all of this. But do you have comments on your approach, the halo tag? Monoallelic, affecting expression levels, affecting other physiology, nuclear, in terms of absolute amount relative to untagged.
Okay, so that's a really good question. So basically, I did check after the tagging, so I did check what zygosity it is. So it is homozygously tagged. And throughout the whole process of generating the line, because it's like selecting for the best clone and everything, and what is a best clone. So for me, it was the one where I could see a proper relocalization upon cell density changes. But what I did do Westerns against the parental line, the wild type MDCK, and the levels seemed the same. It was more that there was an upper shift of the YAP, obviously, because of the tag. And
say that again. There was just a molecular weight shift.
Yeah,
that's all you mean.
Yeah,
I see. Yeah,
the Western because of the tag, right? So it's
yeah.
But other than that, it seemed like the levels seemed to be similar, protein level wise. But I didn't go down to, I don't know, looking at the transcript level, this sort of stuff. I didn't check that.
No, they did that. If you're unaware of that work, it's good, because I was just at a conference, Biophysical Society conference, and halo tags being done, for example, on piezo, and I asked the same questions, and as opposed to TD tomato that's already out there, TD tomato piezo just as I'm hearing complaints. It's like, well, it doesn't have the right levels, and I said, yeah, that resonates with the Allen Institute. This may be the better way to go with the endogenous, right? That you're not going to affect any sort of transcript, feedback loops, as well as localization, phosphorylation, with presumably a smaller tag here, right? This is a much smaller tag, less to get in the way, right?
I mean, it's still, well, it's sort of, I think, almost the same size as GFP, or slightly bigger. Like it's still a sizable tag, but
is it? I missed that. I thought it was much smaller. Okay, yeah.
I don't think it's much smaller.
Okay, all right, very good.
The advantage with the halo tag was that it was brighter than the other tags. Like we did try cherry, but it wasn't bright enough to like see when selecting for the cells. And also, like, there are different modalities, right? So with the halo tag, I can then use the halo pro tag, which is something I've been working on, and I'm now still analyzing the data. So I have done also, since it's a homozygous line, I could induce the degradation of the entire YAP in the system and see what happens.
Other advantages then. Okay, thanks a lot.
So there was a bit that too. Thank you.
Yeah, Madhura, do you want to unmute?
Yes. Hey, Windie, that was excellent. Your videos are amazing. So I just had one question for that last part of your talk where you were talking about the cells that didn't have the ECM substrate. Does the difference in YAP localization, like, does that kind of reflect what might happen in sort of free-floating cells in the body, like immune cells?
That's an amazing question. I really didn't think about it, but I mean, I didn't really ever look at immune cells and the regulation of YAP. I'm sure there is plenty of literature where we can check that. But what I do know is that when people in general, like initially back in the days, wanted to understand how focal adhesions affect YAP, they did this really simple experiment where they would plate cells, like single cells on a substrate or like in the wall plate, and then just add trypsin and see how does the YAP localization and the regulators like phosphorylation and stuff change. And there they saw also that like when you have trypsinized cells that are like free floating, a bit like what you said with the immune cells, YAP is also cytoplasmic. So it does recapitulate a bit what has been shown in the literature with single cells. Does that help?
Okay, yeah, no, that totally makes sense. Thank you.
No worries. Thank you for the question.
We have a couple more questions in the chat. Niranjan?
Hi, very nice talk, Windie. One question is, in your future work, you suggested that ECM remodeling might modify the YAP localization. Do you have some ideas on how you can test with the kind of assays that you have?
That's a really good question. So one idea I had, obviously, so I don't know how you can really do induce ECM remodeling as you go and image, but one idea I had, if it is truly really ECM dependent, was to also look at redoing the system with the stretching, but on a different ECM. So instead of growing cells with the deformable substrate with collagen, what happens if we add laminin? Because from initial studies I know, and also from work from the Perirukas lab, if you plate cells on laminin, YAP is more cytoplasmic. And I also saw that in my system. I haven't shown due to time constraints today. So I know already that in this ECM, maybe due to the specific integrins that interact then with the laminin, YAP is more cytoplasmic. So if I put this on, I would suspect that even stretching might not lead to YAP nuclear localization. So that was another negative control I was thinking that could have been a good future work.
Nice, thank you. And one last question. Do you have comments on what is the mechanism by which this relocalization happens? Is there any molecular motor based transport?
So for now, it's really hard. It's a big question in the YAP field, I feel. So there are some people who say, okay, when you stretch cells, then what happens is that the nucleus becomes slightly more flatter, and this leads to more opening of the nuclear pore, and then YAP can go in more. This could be a hypothesis. So this is something I need to more carefully analyze. So now that I have the data, my next step is really to look at like the changes in the nuclear shape and see if it correlates with anything. But I think it could also be just that the stretching somehow seems to just promote the downstream focal adhesion signaling I was talking about, the SAR kinase, that it's induced by stretching, and this in turn promotes more YAP phosphorylation at the site that promotes it to be in the nucleus. So it could be that just too.
I see. Okay, thank you so much, Windie.
No worries. Thank you.
We have another question in the chat box. Jay Yasha Kresh. I don't know if he's still here. I can also read the question. Great talk. Is it oversimplification and or wrong to say that the cell-cell and cell matrix mechanotransduction behave like CSK force balance, rheostat in regulating the YAP signaling?
just touching. What do you mean with CSK? Does anyone know?
I don't know.
With rheostat, so the way I'm just going to interpret the way I read. I understand
Yeah,
the question.
sure.
What do you mean as in like, do they work as in like a seesaw? Like if one is higher, the other one is low. Is this how I should interpret the question? If it is, then I
Probably
would say,
cytoskeleton. That's what the CSK is, probably cytoskeleton.
ah,
Yeah.
Thank you.
Cytoskeleton, yeah.
I'd say yes, sort of, like in the sense that like with the cytoskeleton, that like depending on if it's different or not, it affects the up. Then yes, I think that the cell and cell matrix also have this thing that depending whether or not they're more active or not, they are the one that is more dominant is the one that affects the up the most. So in cell growth, for instance, it's more the cell junctions that take over the role because there are more of them. And as the colonies grow and there's less space for the individuals cells to feel at the bottom, the basal level, the ECM, this makes them the more dominant one. So, but that's why in my title of the talk, I say epithelia, because it could be different in different cell types, I'd say. Like if it's like fibroblasts or mesenchymal cells, they could behave completely different.
I see. Charlie has his hand up.
Thank you. That was a great talk. I really enjoyed it. And I was kind of curious about the three-dimensional localization of YAP, whether it localizes evenly along the Z-axis in these cells, maybe even as they're undergoing transitions like the epithelial mesenchymal transition, and whether it's sort of enriched at contact points either with other cells or with the extracellular matrix at the basal surface, and the extent to which it localizes at the whole side of the cell.
That's an amazing question. In fact, in my PhD, I spent a lot of time to make sure that we really capture the 3D localization of YAP. So in general, in the NDC case, as shown already in the immunostains from the previous work from people, there's this really mixed hazy localization. So epithelia in general, if they're very epithelial, the cells, the YAP localization is never truly very, very nuclear than what you see in like mesenchymal or fibroblast cells, which is a bit of a challenge then how to quantify this. So so for me, the YAP localization was always like what you said in the Z-axis a bit everywhere. And it was more to see whether it's more concentrated in the nucleus or not. So to check this, what I did is basically with the help of a former postdoc from the Guillaume Salbreux lab, Matt Smith, he developed a program where basically you can create a height map of the Z stack of the nucleus. So so I can align all of my Z stack for the YAP to be based on the focal plane of the nucleus to see what is inside the nucleus and what is not, if that makes sense. So what we did, we didn't look at above and below to be sure that we can just see how much is in the nucleus or not. Because if we start taking everything, it's a bit like doing projection on Fiji. You wouldn't be able to really assemble what is going on. But it's due to the nature of the cell line. If we were using maths, it would have been a whole different scenario. And no, I haven't seen any like enrichment of YAP in the cell-cell context or on the focal adhesion. So it never really was somewhere specific except in the nucleus when any like specific stimuli was put on.
Great, thank you so much.
No worries.
And Guillermo has his hand up as well.
Yes, so in your stretching experiments with like PDMS and then matrix and then cells on top, and then you see the nuclear translocation of YAP. My question is, can you push the cells to a mesenchymal phenotype just by doing this stretching?
No, they are very epithelial.
So
Just by stretching,
if you do like cyclical stretches, right? If you keep doing that to a certain point, I mean, I guess I would. How stable is this nuclear translocation of YAP following the stretch, right? How soon it comes back to the cytosol, and then if you would give like enough stimuli, enough stretch to the cell that would keep YAP for enough amount of time inside the nucleus, now would you be able to push the cell to a mesenchymal phenotype? You see what I mean?
Okay. Yeah, I see what you mean. I haven't looked that far because I only stretched for like a bit more than an hour and then stopped the experiment to go to the next monolayer. So I'm not sure what happens afterwards. But that's a really good question. But I think just because working with these MDCK cells, they're so epithelial, I think it will need a lot of stretching and cyclic stretching for it to turn into something else, if that makes sense. I don't think it, especially with the stretching system I'm using. If it was based on like the ones, the commercial ones where you have the PDMS and you can easily like get it to do it over like 48 hours maybe, but this system, I don't think you can leave the monolayer for that long. But it's a really good question. Thank you.
Okay, I think that was really a great talk, and we had a great discussion. Thank you so much, Windie, for the talk.